Hello and welcome back to the Biochemistry for Health Sciences channel. In today's video, we shall continue the series Understand Not Memorize Metabolism, and we'll discuss specifically the pyruvate dehydrogenase complex or PDH. In the previous videos, we discussed why this conversion of glucose to ATP, basically the generation of ATP from glucose. So this pathway that we call main street, why this main street is perhaps the most important metabolic pathway in the human body. So main street is composed of glycolysis, the PDH complex, Krebs cycle, electron transport chain coupled to oxidative phosphorylation. And in the previous video, we talked about the first part, glycolysis. And today we shall focus on the second part. So in the previous video, we talked about this part here, glycolysis. And today we shall focus on this part here called the pyruvate dehydrogenase complex. So what are the most important features of the pyruvate dehydrogenase complex? The pyruvate dehydrogenase enzyme is really referred to as a complex, a PDH complex, because it's a multi-enzyme system, and it's made up of basically three enzymes that are joined together, E1, the E2. Now the E2 has a swinging arm that is shown here. So this is the E2. And then there is a third component to this multi-enzyme complex that we call PDH, and that is E3. So what is the reaction that this complex catalyzes, the PDH complex? Here is the reaction. So basically the substrates are pyruvate, coenzyme A, and NAD, and the products are acetyl-CoA, carbon dioxide, and NADH. This reaction catalyzed by the PDH complex is a redox reaction. More specifically, it's an oxidation reaction. NAD is an oxidizing agent. So basically, pyruvate is being oxidized. This reaction is also a decarboxylation reaction. So what is a decarboxylation reaction? It's a reaction where the carboxyl group of a substrate comes out as carbon dioxide. Okay, so if you remove this carboxyl group, what we have left is this, which is this part here. And this carboxyl group comes out as carbon dioxide. So that's called a decarboxylation reaction, where the carboxyl is lost as the carbon dioxide. So basically the pyruvate carboxyl is lost as carbon dioxide, and we get an acetyl group. Since we are talking about main street, remember that the first part was glucose getting converted to two pyruvates in glycolysis. So basically in main street, this is the first reaction. Since there are two pyruvates here, we get two carbon dioxides produced. So this is the first reaction in main street where the glucose molecule is losing two of its carbons as carbon dioxide. Since this reaction is oxidation as well as decarboxylation, so there is oxidation and also there is decarboxylation, we can combine these two words and say that this reaction that is catalyzed by pyruvate dehydrogenase complex is called an oxidative decarboxylation reaction. So there are a few reactions in metabolism that are oxidative decarboxylation. This is one of them. Another very important thing to note about this reaction, under cellular conditions, this reaction is irreversible. So note the arrow is only drawn in this direction. The arrow is not drawn reversible. Okay, so under cellular conditions, we can convert pyruvate to acetyl-CoA, but we cannot convert acetyl-CoA to pyruvate. Okay, and this becomes very important later as we talk about carbohydrate metabolism as well as lipid metabolism. So this is an irreversible reaction. Now we are always interested in the energy aspect of a reaction. This reaction does release NADH. So NADH is a product. So if you go back here, you can see that NADH is a product. And NADH, this reaction is happening in the matrix. So the pyruvate dehydrogenase complex is in the matrix. So NADH is released in the matrix. And this NADH can enter the electron transport chain oxidative phosphorylation system and generate ATP. So each NADH can generate 2.5 ATPs, so 2 NADH from a glucose can potentially give you 5 ATPs. So since this reaction produces NADH, since NADH is a product of this reaction, you can get ATP potentially from this reaction. And since this NADH is produced in the mitochondria itself, not in the cytoplasm, like this NADH here is produced in the cytoplasm, but this is produced in the mitochondria, so this doesn't need any shuttle. It can directly enter the electron transport chain. So no need for shuttles like in the case of this cytoplasmic NADH. Now this very important reaction, the PDH complex reaction, requires the help of many coenzymes. Okay, many coenzymes. FAD, for example, is derived from the vitamin B2 riboflavin. NAD is derived from niacin or B3. Coenzyme A is derived from pantothenic acid or B5. Thiamine pyrophosphate is derived from thiamine or B1. And there is the lipoamide coenzyme that is part of E2, which we'll see in a few seconds. So clearly, if one is deficient in any one or more of these vitamins, it is going to affect this reaction. So let's look a little bit more closely at this reaction catalyzed by the PDH complex and the role of the coenzymes in this reaction. So in the first step, the pyruvate is decarboxylated. So the carboxyl group of pyruvate comes off as carbon dioxide. This is catalyzed by the E1 part of this PDH complex, and TPP is required to do this decarboxylation. And that's because this pyruvate is an alpha keto acid. Okay, so decarboxylating an alpha keto acid is very difficult, and we need the help of the coenzyme thiamine pyrophosphate. So what's an alpha keto acid? So here is the acid group. This is the acid group, and this next carbon to the carboxyl group is the alpha carbon, and the alpha carbon is a ketone. So pyruvate is an example of an alpha keto acid. So anytime in metabolism when we have to remove the carboxyl group from an alpha keto acid, we require, or the enzyme requires, the coenzyme thiamine pyrophosphate. So here is the structure of thiamine pyrophosphate, and you can see the two phosphates joined together to form the pyrophosphate. So once the carboxyl group comes off as carbon dioxide, what we have now is the acetyl. So in the second step, the lipoamide, you can see here the disulfide part of the ring of lipoamide connected to E2 reacts with this acetyl, and basically the acetyl group is combined to the lipoamide. So at this stage, the acetyl group is now attached to the lipoamide swinging arm. Now the arm will keep on moving, and in the next step, the acetyl group will be transferred to the coenzyme A, and this will form acetyl coenzyme A. So you can see the acetyl group here now is part of coenzyme A, and that will give us acetyl-CoA. So at this stage, we see that the pyruvate has become decarboxylated to give you acetyl. The acetyl has combined with coenzyme A to give us acetyl-CoA. So we basically have these products here, acetyl-CoA and carbon dioxide. Now let's look at the role of the NAD that becomes NADH. So note that the lipoamide arm, the ring, has become thiols, 2 sulfhydryls. Okay, 2 sulfhydryls. Originally it was a disulfide. The ring was a disulfide. Now, but after this reaction with the pyruvate, with the acetyl, it has become reduced to 2 SH group, 2 thiols. So basically, in the next step, the enzyme will have to reconvert this reduced form into the oxidized disulfide form. Okay, so that's where the next step happens, and that involves E3. So the enzyme E3 will now oxidize these thiol groups, these sulfhydryl groups, into the disulfide, bringing the lipoamide arm back into its original chemistry, original structure. This oxidation of the sulfhydryls into the disulfide requires FAD. They said FAD that's actually attached to the enzyme, to the E3 part of the enzyme. So this is actually covalently attached to the E3 part of the enzyme, and it also requires NAD. Now NAD, on the other hand, is not attached to the enzyme. NAD actually comes into the active site, and after its oxidation of the thiols into the disulfide, NAD becomes NADH, which is a product, and that gets out of the active site. So the actual oxidation of these thiols into a disulfide requires NAD and FAD, and the mechanism of this reaction is still poorly understood. So that is the reason why FAD is not seen in this reaction, the oral reaction, the role of FAD, because FAD is covalently attached to the enzyme, and it basically is helping the NAD coenzyme to oxidize the thiols into the disulfide. So NAD, you do see NAD comes to the active site, and then after the reaction comes out as a product. So this is not typical, as you will see in many other reactions. FAD is a coenzyme, but the FAD will come out as FADH2. So you do see FAD as a substrate and FADH2 as a product, but not in this case. Okay, so this can be a little confusing for students, but that's the reason why we do not see FAD in the oral reaction. So PDH is a complex, a multi-enzyme complex, and we'll see in the human body later in metabolism there are other multi-enzyme complexes. These multi-enzyme complexes are much more efficient than having individual enzymes. And why are these multi-enzymes more efficient than having, let's say, 3 individual enzymes, E1, E2, E3? There are a few reasons for that. One is that there is an increased frequency of substrates colliding with the enzyme. Remember, reactions happen by collisions. So the substrates are all nearby one another, near the enzymes. E1, E2, E3 are all near one another. So all this there's an increased chance of a substrate colliding with the enzyme because all of them are close to one another. So that increases the reaction rate. The second reason is the intermediates that are formed, they don't react, there's a less chance of them reacting with other cellular molecules because the intermediates are very close to the next enzyme. And finally, these three enzymes being very close to one another, they can be more precisely regulated, more finely regulated. Another very important function of the PDAs is that this reaction, the conversion of pyruvate to acetyl, this reaction connects carbohydrate metabolism with lipid metabolism. Now remember, as we mentioned earlier, carbohydrates such as glucose is probably the most important fuel for energy generation, and the second most important are fatty acids that are lipids. So basically this reaction connects two very important fuels that are used by the human body to generate energy. So we saw earlier how glucose can be broken down into pyruvate by glycolysis, and in the future we'll see how pyruvate can be used to make glucose by gluconeogenesis, and this becomes very, very important during starvation periods. In the future we'll see how acetyl-CoA is a very important building block for making fatty acids as well as cholesterol, and we'll also see how fatty acids, when they are broken down, they will give you acetyl-CoA. So this connection basically means that we can take glucose, so we can take carbohydrates because that glucose will be broken down to pyruvate, and that pyruvate can be converted into acetyl-CoA, and that acetyl-CoA can be made into fatty acids, into lipids. Okay, so this reaction is only in one direction, so humans can easily do this. We can take excess carbohydrates and make it into lipids. However, we cannot take fatty acids and make it into glucose because this reaction goes only in this direction. So we cannot do the reaction in this direction. So this cannot happen. So humans cannot make lipids into carbohydrates. We cannot do that. One reason is because this reaction is irreversible. It can never happen in this direction. The second reason is we just don't have any other mechanism or any enzyme to make fatty acids or lipids into glucose. So this is a very important point that we shall elaborate on further when we talk about starvation states that humans get into. So these are some of the more important aspects of the PDAs complex. In the future video, so we've already talked about this part of main street. In the future video, we'll talk about the third part, which is the Krebs cycle. So until the next video, have a wonderful day. We'll hopefully see you soon in the next video.